Hey,
Hello, how are you?
I'm good. How are you?
I'm good.
Cool. All right, what's new?
Um, Not much.
Mhm.
Um, I took blueprint full length 3 yesterday. I didn't take like any car section, so there's that. But I got a 126 in Chem Phys,
Mhm.
128 in biochem and psych soc.
Very nice. Did you not do a car section because of like you didn't know like which car section to do, or did you feel like you're already good with cars? Like what?
Um, I feel like I definitely still want to keep practicing cards. I just didn't take it, but I uh normally I would have done like the AMC deck from that.
Got it. Okay. So cool. This is good. Um, so awesome. Okay. Yeah. How did you notice? So how was like the timing? How was like your stamina and all of that stuff?
Yeah, the timing was better. Um I felt like at least like that 1 point increase in Chem Phys might have just been not a content increase, but just because I got through more questions increase. And for biochem, I was also surprised that I had around 7 minutes that I could review like the flash questions at the end.
Hmm.
Yeah. I did notice that I was sort of like, because I knew, like, I know myself and I know that I would have wasted a lot of time in the like calculation questions. So I ended up skipping most of those questions, which were a lot for like the blueprint pulling 3. Um, but I think in the end, like, it was better to just like flag them and like if I had time in the end, go back and do it.
Hmm.
Mhm.
Got it. Okay. So I'm trying to see where I wrote the okay. Okay. So I in like your spreadsheet, there's like the MCAT score that you had, and then the blueprint full length 5, and I'll just add the blueprint full length. Oh, it was 3 this time. Okay. So, and this was just yesterday, right?
Yes.
Okay. Okay. Um, yeah, okay, cool. Yeah. So your CP score went up by 1 point. BB is constant. Psych Soc went up by 1 point. So that's pretty good. Did you get a chance to review it?
No, I haven't.
Okay.
But I did identify two passages in Kempis that were like that I got a lot of questions wrong that we could probably review those.
Yep. We could do that.
Yeah, it's kind of tough for me to try to do the blind review before because I take them on Saturdays and
Mhm.
then meet on Sundays. Um, yeah.
Yeah, we could probably do something where we, I guess, um, go over like the previous test when we meet, like prior to, um, but yeah, I think it's okay. We could still make most of our session.
Yeah. Oh, that's good. I guess I could take that into account next time.
Hmm.
All right. Yeah. Just pull this up. So I sort of did like let me like as if I were retaking the test in sections.
Yep.
Um, Okay. So it's passage 3 and 4 that I got a lot of questions from.
Okay.
But we could start here. Mhm.
All right. So
Uh,
so why don't you
yeah, I can start the
yeah
read.
uh slowly, and then you can um kind of tell me what they're saying in each sentence in your own words.
The pH sensitive genetically encoded fluorescent biosensor known as Y32 is capable of sensing a cell's ATP to ADP ratio, an important indicator of its metabolic state.
Okay. What are they telling us here?
So they have a biosensor named Y32 that can detect or like sense the ATP to ADP ratio. So like whether it's
Mhm.
like energy level, energy state,
What's
pH sensitive.
what's fluorescence?
So light. So it's emits light.
Uh-huh. So I'm not sure if we went over. Did I go over before with you, like, what if like a photon gets absorbed by an atom?
We went through like electron capture and
Oh, like the radioactive decay, but we never went over just like a photon getting absorbed, right?
no.
Okay. So, um, okay. Okay, let me just go over that real quick. So let's say that we have an atom with, you know, whatever. Let's say like this is the nucleus here, and then that's an electron in this shell, which we can call n is equal to one shell. And then we can have another shell around that, and I'll make one last shell around it. So let's say that this is the n is equal to one shell, and then n is equal to 2 over here, and then n is equal to 3. So, um, and then let's say that the energy level difference between the n is equal to one shell and the n is equal to two shell, let's just say it's 1 electron volt. And then let's say the difference between 2 and 3 is 0.5 electron volts. Now, let's say that I shoot a photon with 0.99 electron volts at this atom. What
Mhm.
would happen?
Um, so you don't have enough energy to excite the electron to like to 1 to 2, right? But I guess what is 0.5 ev you maybe could excite an electron from like 2 to 3.
Uh-huh. Okay,
Yeah.
so basically, all right, what if I had two photons with 0.99 electron volts?
um I'm trying to think of like quantized energy. Um, and like
Mm-hmm.
I feel like you would still only have like the 2 to 3,
Mm-hmm.
not the 1 to 2.
Yeah. So we start with the ground state at n is equal to 1. Um so yeah, we can't like, skip to like 2 to 3.
Oh, I see. I see. I thought you can't do that. You can't like isn't there's no other electron in like the second energy level that you can excite to a third?
Uh Not in this example, I guess, but no, in general, yeah, you want in this type of like topic, you want to start off with the electron being in the ground state.
Okay.
But okay, good. Very good. Most people, think that, they can be additive and stuff, but they can't be additive. It's all or nothing. And that's what the quantum in or quanta in quantum mechanics is about. So something that they like to ask is like, let's say for this example with 0.99 electron volts, they ask something that they like to ask is what if you increase the intensity? Would that make a photon get absorbed?
So intensity is just having more photons, right?
Excellent. Okay, good, good. So yeah, so would that do anything?
No.
Okay, good. So all right, let's have okay. A photon with 1 electron volt now.
Um, I feel like you would get excited. Um, if you had any extra, I don't know, things are coming back to me. So I'm remembering like if you have extra energy, there is a possibility of like expulsing, like ejecting the electron, and it would like translate into like kinetic energy for that.
Yep, yep.
Does this happen here? Like if not with one, but like if it were higher?
Yeah, yeah. So okay, yeah. So I'll get to that. So this 1 electron volt photon should get absorbed now, right? And so it'll get absorbed, and then to, conserve energy, um, an electron in the atom has to get excited. So it'll get excited to the n is equal to 2 shell. So, it'll go here. And if it's in a higher energy level, it's less stable. So eventually it'll go back down to its ground state. And when it goes back down to its ground state, it will then emit a photon with that same energy level.
I see. Yes. There's an equation for this, and I remember there was a question that I needed to use it, but I couldn't remember it.
Uh-huh.
Um,
Uh, might be something like with like n2 squared minus n1 squared Rydberg type of, could be something like that.
yeah, I think so. I think.
But yeah, so okay, so that's so there's three scenarios. So this is the first scenario where energy is just conserved. Um, the other scenario that you're thinking of is, well, I guess I'll do that last. So for the second scenario, let's say that we have, I'll use green. So let's say that we had a photon with 1.5 electron volts. What would happen now?
So you can go from one to
Mhm.
two and then to three.
Yeah. Oops. Yep. So it'll go from 1 to 3. So, this can go all the way there. Okay. Now, if this molecule has lots of conjugation, right? Um, so what does that mean? That means that it can undergo resonance stabilization. And since resonance stabilizes something, a molecule that has a lot of conjugation can, through resonance stabilization, can enable an electron to relax back to its, let's say, n is equal to 2 shell without emitting a photon. And then eventually, when it goes from n is equal to 2 to n is equal to 1, it will emit a photon, except the photon that it emits will have a lower energy. So, um, so in this case, it'll absorb the 1.5 electron volt photon, exciting an electron to its n is equal to 3 shell. And through resonance stabilization, the electron will relax down to its n is equal to 2 shell without emitting a photon. And then eventually when it goes from n is equal to 2 to n is equal to 1, it will emit a photon, and that would be with a photon that's 1 electron volt. So this is what happens with fluorescence. So in fluorescence, what you have happen is the energy of the photon absorbed is larger than the energy of the photon emitted. And you could also express this with wavelength, like the wavelength of the photon absorbed will be less than the wavelength of the photon emitted.
Yeah.
Mhm.
I'm thinking of fluorescence in this case, like would this mean that you would need like a conjugated system in order to have fluorescence? So
Yeah.
you would like a ring or like some like double bond, single bond, double bond structures.
Geez. Okay. Uh, yeah. So that's why, if you ever look up the structure of like melanin or chlorophyll or some dye, right? Or a pigment, they're going to be full of there's various ways to say it, but you'll see like either like they'll say conjugation, extensive conjugation, resonance stabilization, they could say electron delocalization, they could say pi bonds, they could say um yeah, all of these things that mean the same thing ultimately. So yeah. So okay, so that's fluorescence. And then the last example is what you kind of touched upon with so let's say that we have um well, okay, let's say that I add an n is equal to 4 shell, and the energy difference between 4 and 3 is, let's just say, I don't know, 0.3 electron volts. So that essentially means from 1 to 4, that's 1.8 electron volts, right? So let's say I shoot a photon with an energy of 1.8 electron volts. It will cause um it to, and like let's say that the n is equal to 4 shell is the outermost shell. Um yeah, if a photon with 1.8 electron volts gets absorbed, it will excite that electron up to its outermost shell, and it can be unbound at this point. Um, Meaning that it's kind of like, well, the atom gets ionized now because an electron is unbound. Um, but the electron's not moving yet. And so this is the situation with the photoelectric effect, where, as you said, the kinetic energy of an electron is going to be equal to the energy of the photon that's absorbed minus this thing called a work function. And the work function is this Greek letter over here, which is essentially like uh it's essentially like the ionization energy of something. So like in this example, let our work function is going to be the 1.8 electron volts, because that's how much energy it takes to, you know, make an electron unbound. But it doesn't have enough to, or an excess, so the electron isn't going to be moving. But if we had shot photons with, let's say, 2 electron volts, then we can use this formula to find out, oops, to find out the kinetic energy of the ejected electron. So this would be 0.2 electron volts, and that is now going to be the kinetic energy of an electron that is not just excited, but it's ejected. It actually is leaving the atom.
I see. Yep.
Yep. So yeah, knowing so it seems like you have a good grasp on this, so that's good. Because it's like super high yield. So yeah, so good job. Any questions?
Nope. I think this was useful. I guess for the fluorescence, the
Mhm.
part of it going I guess it's more stable, but I don't really quite get why it doesn't emit any energy when it goes from 3 to 2.
Uh-huh.
Like
Yeah.
what?
So another way that they might describe it is like it's stabilized through like intermolecular forces and stuff. But yeah, but the reason that it doesn't emit a photon as it goes from 3 to 2 is because it's stabilizing the excited electron through its own like resonance with so if it's stabilizing it through something like that, um it's not going to release a photon because the entire idea is that a photon has energy, and if you absorb that photon, you need to express that increase in energy by having an electron get excited. Um, Eventually, the electron will relax back to its ground state. So, if you absorb a photon, you excite an electron. If an electron relaxes, it'll emit that photon. But in fluorescent molecules, yeah, you're just going to have, due to the resonance stabilization, it'll stabilize the molecule overall to let it relax down to a lower shell without needing to emit a photon.
I see.
So, so yeah, those are the three types of examples, and you did well with, you right away you knew that adding or intensity is not going to be something that really does anything here. So, okay, great, great. So yeah, so we have a fluorescent biosensor, and it can sense a cell's ATP to ADP ratio. It's pH sensitive. Okay, you can continue.
Um,
Want to take a screenshot of this?
I took notes, so it's good.
Sure. Okay, so I'm gonna clear it.
Mhm. Yes, so the biosensor is a single gene product that contains a sensing domain and a reporting domain. Because could we talk a little bit about a single gene product? So does this mean that um that it's not, does it like not undergo like alternative splicing or things like this? Oh, yeah.
So, um, yeah, I mean it'll still undergo that, but um, I think what they're or the reason that they're saying this is that, well, the main difference between so like eukaryotic cells can have um mRNA that's like monocistronic, like coding for just a single thing, whereas
Mhm.
prokaryotic cells like bacteria have polycistronic mRNA, which codes for more than one product. So I think that they're trying to tell us that either, well, I guess like two things. They're trying to tell us that it's not like made in bacteria. But it could also be telling us uh that the biosensor is made like the entire biosensor is made at once. It's not like some subunit from some other gene product can mix and turn into something with, you know, uh, this gene product. So I think that's just what they're saying. So, but as opposed to one gene coding the sensing domain, one gene coding the reporting domain instead of something like that.
I see.
Like,
So means sensing domain and a reporting domain. Okay. Uh, these domains are physically coupled such that Mg to magnesium 2 plus ATP binding causes a conformational change in the sensing domain that is transduced into a conformational change in the reporting domain, which is a chromophore concentrating photovoltaic module. Okay, let's take this back.
yep.
Okay, so the biosensor has two domains. Is this like two different subunits when it says domains?
Uh, we don't know yet. Um, so yeah, so I would say we don't know yet,
Okay.
but yeah, we know at least that they're just different, domains. But yeah, I would say that we don't know yet. Um, but yeah, continue telling me what
Yeah. So it seems that like magnesium binding, so like so a ligand binding causes like a mechanical change. So like a it's like a ligand binding
when you say
kind
when
of receptor. Huh?
you say mechanical change,
Yeah.
you mean that by that?
Like it's I'm thinking of like this binds and then it causes stuff to move and then cause a change. Uh, is that not mechanical? I don't know.
Uh,
Uh,
I think yeah. I mean, I think mechanical usually just has like the more like a physics kind of connotation, but in the sense that something is, some type of kind of like a tool of some sort, but conformational change, I would say, is just change in shape,
yeah,
right?
yeah.
So, okay, so that can happen. So, um, yeah, so the domains are coupled, and
Uh,
if yep, go on.
um, and then like so one of the domains causes elicits like a conformational change in the other, which has the chromophore this. So the chromophore, I'm thinking like the fluorescence. Like this is probably the part that's photovoltaic module. So this is probably like the part that is like producing light or that we sense.
Mhm.
Um, and it's called the reporting domain. So I think that matches.
Yeah, so okay, so they're telling us that um okay, so they're telling us that magnesium ATP binding causes, so I'll make like a proportionality, causes conformational change. I'll just write change in the sensing domain, which causes a conformational change in the reporting domain. And the reporting domain is the CPV thing, the this photovoltaic module. But but yeah, I would start off like this. You know, magnesium ATP binding causes a conformational change in the sensing domain, which causes a conformational change in the reporting domain.
Yes. And this can be monitored with microscopy.
Oh yeah, the transduced.
Oh, I forgot that part. The transduced perturbation causes a change in the optical signal provided by the reporting domain, which can be monitored with microscopy.
Mm-hmm. Yep. So essentially, that conformational change in, so when they say the transduced perturbation, which conformational change are they talking about?
They're about this, when this binds and causes a conformational change.
Um, is it the conformational change in the sensing domain or the reporting?
Uh, um, I thought it was a reporting one.
Mm-hmm. Yeah.
Yeah.
How do you know?
Sorry?
How do you know?
Um,
Mhm.
well, first, because like, isn't the reporting domain what they can monitor? Because that's the one that's like emitting light. And so that's the one that they're able to like view and with microscopy.
Mm-hmm. Yeah. So it also says here reporting domain, but what I wanted you to see is just that they mentioned this, the transduced perturbation, and here we see that this binding can cause a conformational change in the sensing domain that's transduced into a conformational change in the reporting domain. So those are just the words that, give us that information. But okay, cool. So okay, we got that. Um, yeah, we can continue.
The biosensor can bind both the magnesium ATP and ADP with very high affinity, 1 micromolar. In the cytosol of a normal cell, the concentrations of ADP and magnesium ATP range in the hundreds of micromolars and approximately 1 millimolar, respectively.
What's KM?
The concentration of substrate to reach like half the Vmax.
Good, good, good. So is it proportional to binding affinity or inversely proportional to binding affinity?
Um, I would say that it's like binding affinity, but like a lower one would mean that it's a higher binding affinity. Um, is there like a symbol for binding affinity? I just have never really used
Uh,
that.
Not really. But yeah, the it should be an inverse proportion. Um, so okay, cool, cool, cool. And then they tell us that, well, yeah, the biosensor can bind both of these with high affinity. And then in the cytosol of a normal cell, the concentrations of ADP and then the magnesium ATP range in the hundreds of micromolar for ADP, and then approximately 1 millimolar for the ATP, right? The magnesium ATP.
Yes.
Um, which is larger.
The 1 millimolar?
Good. Good, good, good. Okay. By like a factor of like 1,000, right? Roughly. So, okay, cool, cool, cool. So okay, so we see that there's significantly higher amount of the ATP. Okay. Um, continue.
As a result, there is competition for the single biosensor nucleotide binding site that allows the biosensor to detect the cytosolic ratio of ATP to ADP. So they both can bind with equal affinity. Of course, there's one has a higher concentration, so like probably more probability of being bound. But they're both competing to be on that active site.
Mm-hmm. Yeah, the competition part, you know, sounds a little bit about like competitive inhibition, even though, we don't really see that explicitly stated yet, but we want to think in those terms. So So yeah, since there's competition for that binding site, it means that the biosensor can detect the ratio of, the ATP to ADP. So, okay, cool. You can continue.
The Y32 fluorescence excitation spectrum contains two peaks because CPV can exist in two charge states. So it has an A state and a B state, and the difference is the proton, like the hydrogen.
Hmm? Yeah, and you can see how this is a heavily conjugated molecule, right?
Yes.
So that's what we expect to see for fluorescence. Good, good. So, okay, continue.
The protonated chromophore, called the A state, is favored in the ADP bound state, bound conformation, and the deprotonated chromophore, which is called the B state, is favored in the magnesium ATP bound conformation.
Mhm. Yeah, so now I would write perhaps something like A state proportional to ADP bound, and the B state proportional to like the ATP, like whatever, I'll add the magnesium too. That's bound. Good.
The fluorescent intensity produced in a cell expressing the sensor is proportional to the number of sensor molecules excited.
All right. So I would add that too. I would say, fluorescence intensity proportional to number of sensor molecules that are excited.
Yep.
Okay.
The difference in fluorescence between the magnesium ATP bound biosensor and the ADP bound biosensor peaks is seen in their fluorescence excitation spectra. The spectral ratio of the peaks of the excitation spectrum directly reflect the cellular ATP to ADP ratio.
Okay. So what can you tell me about this figure?
So I have fluorescence on the Y, excitation wavelength on the X. Um, so
Mhm.
it's a fluorescence intensity. This is excitation spectrum. So this is, they are like shining light on it, like are putting energy in, and they're measuring whether they get fluorescence or not. So like whether they are able to,
Mhm.
yeah, I'm not quite clear on like of what we discussed, like what is happening. Is it like, because fluorescence, usually it's when it's like going from like higher to a lower energy, right? As in the graphs. Um, but I guess we're seeing here like where the transitions are happening depending on the amount of like wavelength. So like this is like an indication of energy that I'm putting in. And it seems that like for the A form, it's at lower wavelengths. So I would need more more energy to get like the max fluorescence here, versus for the B form, it's at higher wavelengths. So at lower wait, I probably messed that up. Higher wavelengths, lower energy, lower wavelengths, higher energy.
Mhm.
Yeah.
Good. So,
Like energy. Yeah.
mhm. So they're telling us that the difference in fluorescence between these two peaks is something that we see in this figure. And then they tell us that the spectral ratio of the peaks of the excitation spectrum directly reflects the cellular ATP to ADP ratio. So essentially, the peak um, like if I did like the fluorescence intensity at or fluorescence intensity of, let's see, A form is the ADP one. So, okay, I'll do B. Fluorescence intensity of B, which is at like, or peaking at 500 nanometers over the fluorescence intensity of A, which is at 425 nanometers, because they're saying the spectral ratio of the peaks
Mm,
it directly reflects the cellular ATP to ADP ratio, so something like that.
I see. So I guess like in a normal cell, as I mentioned, like you would see two peaks. And here they have it separated, but normally you would see just the two peaks on like one excitation spectrum. And then they would just take the like, would it be the intensity difference at those two peaks?
Mm-hmm. Yeah, yeah, yep. So, okay, great. So we essentially see that um that magnesium ATP binding causes a conformational change in the uh whatever chromophores sensing domain, which can then be transduced into a conformational change in the reporting domain. Um, we know that the overall, I guess, experiment is designed to use, spectroscopy or microscopy and stuff to figure out an ATP to ADP ratio. So we also are being told that the A state is proportional to like it being bound to ADP. The B state is proportional to it being bound to ATP magnesium. So if we take the ratio of the um, intensities for B over A, that would be proportional to the ratio of ATP to ADP.
Yeah, I see this.
Mm-hmm. Okay. Any questions?
Nope. Yeah. So I guess it seems so, but like green fluorescence, I just want to make sure that is like intensity, like the height is intensity here.
Mm-hmm.
Okay.
Yep. The Here I even wrote the fluorescence intensity is proportional to the number of sensor molecules excited.
Okay. Yeah. All right.
Yep. Okay. So yeah, let's try this question out.
So what is the respective hybridization of the imine nitrogen and carbonyl carbon of the CPVB state as it is depicted in the passage? So they're asking about this nitrogen and this carbon, this carbon.
Okay.
So hybridization state has to do with like an amount of groups that are on the like nitrogen or carbon here, or
Yeah.
like bonds or connecting or lone pairs, I think. So let me just check here.
Mhm.
So I think like max divided, this is the part of chemistry I do not feel comfortable with, but like here for nitrogen, like it would want to have uh like five things, at least like five electrons, or so one, two, three. So it probably has a lone pair is what I'm getting at, probably. Um, and then the oxygen, it probably has two lone pairs.
Okay.
Sure. Yeah, I think it has two lone pairs. And I think that you would count the lone pairs as like one group. So like this would be like one, two, and then this would be three. So I'm thinking of like three groups for oxygen and one, two, three groups for nitrogen,
So
maybe?
let's talk a little bit about these hybridized orbitals here.
Yeah.
So, yeah, what can you tell me about these?
Um,
Let's say we have methane here.
mhm.
Um, so this methane is a carbon bound to four different hydrogens. Um, what shape would this be?
Is it like a tetrahedral?
Mhm.
Yep.
So that would have a bond angle of around what?
90?
Um, maybe something else.
Um, what is it?
Mhm.
60? I don't know.
So,
No.
so the the wedges here indicate that it's in, three dimensions. Because sure, if it was in two dimensions, and it's four things that are bound, 90 degrees would make sense. But since it's in three dimensions, it means that each of the hydrogens can be further apart from one another. So the it would be 109.5.
Oh, that number. Yeah.
Yep, yep, yep. Okay, now so that would be the angle between each of these hydrogens because they just want to be as far away from each other as possible. Um, and this would be sp3. Okay. Now, sp2 would be if you have something like, okay, so let's say that we got rid of like,
The hydrogens and had a lone pair. Two of the hydrogens and had a lone pair.
uh-huh, yeah, yeah. So lone pairs will push or repel like the hydrogen atoms and stuff more so than a hydrogen atom would do that. So it should move things like further away from one another. But um like, an easier way of drawing a molecule that's sp2 would just be like something like maybe like I'm just gonna draw like a carbonyl. Um which shows that it's bound, like the carbon is bound to like three things total. And this is a planar like molecule, right? So um the angle will just be 120 degrees, and this is called trigonal uh planar. So that's sp2. And then lastly, if you had, perhaps like let's say like a triple bond like in cyanide here, that would be sp, like just sp, and it will be a linear molecule, so it'll be have a bond angle of 180 degrees. So, there's also like sp3 like d stuff, but for this question, let's just be aware of the different, like what makes sp different from sp2 and sp3. So, okay, now that you have this knowledge, what do you think, you know, what, because they're asked, yeah, what do you think the hybridization would be for the amine nitrogen, which is this one, and the carbonyl carbon, right?
So I think it's SP2
Mhm.
for both. I guess I have a more general question, which I've always been confused. So when it says like hybridization, are we referring to this like molecular geometry and like Vesper theory? Is that what it's referring to? Like yeah. Could you tell me a bit more about that? About like the term of hybridization?
Yeah. So let me see. I think I may have a PDF on it. Let me check. Okay, so I'm putting a link in the chat if you can open that up and share it.
Mhm.
Oh, I
Um,
guess just link to this, but it'll be called hybridization bonding.
should I
Oh,
just?
there it is. Yep. So, so yeah, I talk about it here. So, first we have this linear, like examples of linear molecules, then we have examples of trigonal planar, and you can see how, so the boron tetrafluoride is has identical fluorines bound to it, so it's going to be a precise 120 degrees. But um if you have something like the aldehyde on the right, I wrote that since oxygen is more electronegative than hydrogen, it pushes or repels more. Um, so that just makes the angle between the carbonyl and the hydrogen larger than the 120, and then the angle between the two hydrogens smaller than 120. Kind of like the same way a lone pair would push further. So yeah, and then I drew the tetrahedral, which in 3 dimensions you can kind of see how it's the 109.5 thing. Um,
Yeah.
I drew the shape of the tetrahedron on the right. It's like, a triangular type of pyramid of some sort. Um, and then we have trigonal bipyramidal, which would be like this PCl5, and then the shape again. And then I talk about like the, yeah, look over here. This,
Mm.
I separate the axial ones, like that are just above and below, from the equatorial ones. So, this would make that shape, and then they would make that shape like that. So that's okay. So that's the trigonal bipyramidal. And yeah, let's continue going down. We also now have octahedral. So that'll look like that. And then I wrote, yeah, why do distortions occur? Um, and it's because lone pairs cause greater repulsion. So you could see in this methane again, all the hydrogens are identical, so it's 109.5 degrees. But when we have, let's say, ammonia has this lone pair, and the lone pair pushes, these against, like, it pushes against that stuff, creating like here an angle that would be larger than 109.5, but between the hydrogens it would create an angle that's smaller than 109.5 because of, and that's an example of distortion. um and then and that also explains the shape, like tetrahedral for the CH4, but for the ammonia it's a trigonal pyramidal. And then I have two lone pairs here for H2O, and compared to like one lone pair, it's going to push the hydrogens like closer together and further from the oxygen. So this will be, if this one was 100 and oops, if this one was 107 degrees, this would have to be like less than 107 degrees. So yeah. And then I wrote strength of repulsions. So lone pair, like repulsion is the strongest, followed by lone pair and a bonding pair. Like so lone pair would be like this, right? A lone pair bonding pair would be like this, the lone pair, and then an actual bond. And then bonding pair would just be, between the actual bonds. So, okay. And yeah, let's continue going down. What did I write here? Oh, just these are just distortions of the different ones, of like tetrahedral and all these things. But let's scroll down. I think I have like a chart of, yeah, here we go. So yeah, this chart,
Do
you
you mind like erasing the, or should we keep that?
um
Maybe we should keep it for the rest of the passage.
yeah.
Mm.
But yeah, so this chart you probably have seen before, but it essentially shows you that like the different hybridizations and the different types of shapes or geometries that you can get. And then the and that would be like with zero lone pairs.
Yeah.
Then you want to also start adding lone pairs to get the distortions. So essentially, SP, I drew like a linear structure here with no lone pairs. And you can't really add a lone pair to this. So then we move on to SP2, where you have, this thing with 3 bonds. And then we have, if you add a lone pair here, it'll affect the shape there. And SP3, you have the tetrahedral, and then you have, you know, this thing that's like ammonia, let's say. Um, and then this thing that's like water.
I
There's
see.
also SP3D, which forms this, like bipyramidal, trigonal bipyramidal shape with zero lone pairs, and then you can see how that can adjust to like, this is like T shape. Um, this is linear. This is, I think, seesaw, something like that. But yeah, this just goes into the different types of uh molecular geometries for the different hybridization. So that could be something that you can refer to
Yeah.
yeah, to figure out
Yep.
hybridizations and stuff. Yeah.
Good.
Mhm. Yeah. Okay. So yeah. So good job. Now you see that it would be SB2 and SB2, right?
I see that.
Mhm. Okay.
Um, so question 19, a cell's ATP to ADP ratio is most nearly equal to the ratio of its fluorescence emission intensities when Y32 is excited at. Oh, so they want that ratio. So like when this number is equal to this number, so then it should be in a place where it's equal or like relatively like similar, I feel in here and here. Let's see the options. 500 nanometers versus 420 versus 420. Could be. I don't know. 420. Oh, okay. So I have to take into consideration whether it's like the A form or the B form, I guess.
Uh-huh. So they're asking about ATP to ADP ratio. So what would be
So ATP ratio would be like the B form and like the ADP would be the A form. So like B to A, like I don't know if I'm like thinking about this the right way, but I'm thinking of a spot in these in the wavelengths where like you have similar levels of fluorescence so that they are equal. Is that the right way to think about it?
um you're looking at you're looking, what were you looking at again? Were you saying like where the fluorescence levels are equal, or?
Yes. Like where like they're like yes. Um where the fluorescence levels would be similar, like equal. I guess let me just read here as well. Ground state versus when it is a ground state. What? Oops. I'm confused. um oh, so I think that they just want to know then like what like it's going to be better at the max intensities, right? So that at the max, so like where you have the maximum fluorescence for each. So this is more of like I don't have to think as much as I thought. I think it's more like which one it would be the best readout, like the best reading, like when you program a spectrophotometer to like the perfect wavelength, because it's like at the max of the peak, and that would give you like the more accurate absorbance value. So I think that this is going to be like if it's ATP to ADP ratio, then it has to be like something with 500 and something with 425 or 420, I guess. If it's I guess the order matters here. So I guess 500 to 420. I might go with a um
Mhm,
ground state. What do you mean ground state? I would go with A. Yeah.
mhm. Yeah.
You think?
Oh yeah, I think so too. I think I kind of wrote that here with the fluorescence intensity of B at 500 nanometers over fluorescence intensity of A at 420 nanometers is proportional to the ATP to ADP ratio.
Yeah.
So I would think so as well.
Yes.
Mhm.
Yes. A student theorizes that the differences in ATP to ADP ratios between cells detected in an experiment could be due to variations in Y32 expression rather than differences in the cell's metabolic conditions. Does information support? So it did say here, and I think you wrote it down too, that like this intensity is like about like the number of sensors. So like the number of like these biosensors would matter on what we're observing, I think. Um, so I would say it's in the yes part, but let's see. Y32 fluorescence intensity is proportional to the number of sensor molecules excited. I think so. Let's see. Y32 expression levels can influence the cellular ATP to ADP ratio. I think it's A. I think it's A.
So walk me through the reasoning here.
So the this sensor is like a readout for ATP to ADP levels. But my understanding is that this so you're measuring intensity, but intensity is also like directly proportional to the amount of these sensors that you have around.
Okay.
So I'm thinking that if you have more of these around or less of these around, it might be influencing your perceived ratio, like real ATP to ADP ratios.
Can you select like what I do here, um, and just move it a little down a little bit?
Can I do that? Can I select that?
Yeah. I think on the top of your Zoom like bar or whatever, there could be like a selection tool,
Um, huh.
maybe under like annotate or something. It's annoying because it doesn't let me do it. It lets only the person who's sharing their screen do it.
Maybe I don't know how to do it. I don't see it.
Oh,
Uh, Top share. I can.
I'll just kind of like redraw it here. So at 500.
Annotate? Do I have to click annotate?
Yeah.
And then I can select, yeah.
Yep. That's
Select this
yeah.
and move it. Did it move? No. Interesting.
Yeah, there you go.
Yeah.
Okay, cool. So, okay, let's think about what the question stem is asking us. So, what is it asking us?
So it's someone is theorizing that there might be differences in the expression of Y32 instead of like differences in ATP to ADP that are accounting for like the differences in the readout in the fluorescence intensities.
What do they mean when they say Y32 expression?
Like I guess it could be like upregulated or downregulated. Like it would have like higher there might be more expression of a given like protein in one cell versus another.
So, tell me exactly what you mean when you say expression.
Like more protein versus less protein.
So,
More of the sensors or of the receptors versus less of them.
so that means more transcription, right? Which means more like translation, and that's how you get more of the protein, right? So yeah, so that's called expression, and you'll see this a lot on the test, and you'll see a lot of this type of question where, so the passage tells us that, you know, the differences in, I guess, fluorescence for Y32 uh shows us that there are different amounts of ATP and ADP in the cell. But what can also be the case is that we get a higher amount of fluorescence intensity because we simply have more Y32 to begin with. So so the question's asking us, I guess it's asking us, does the information in the passage support this as a possibility?
Hmm.
Um, Meaning, uh, is this something that they've already, I guess, taken into account? And also for questions or choices like this with like yes and then stuff after it, no and stuff after it. I always like to do like the stuff after it part because yes and no, it's hard to say if it's yes or no because the thing after it is the justification. So that's what I would uh look at. I would look at like, the second part of each. Um, okay. And yeah, so we're trying to see if yeah, so okay. So what are your thoughts then?
Mm. I guess I have two thoughts. I'll try to be think of the no now because I sort of already thought of the yes, but maybe now I'm second guessing myself. Like it is a difference in being so you have ATP to ADP ratios. So and this it's like the same sensor molecule that's giving you this information. Whether it's in like two different states, two different conformational states. Um, and so even if you had 100 of the same sensors, you would still have like these two states so that are influencing your um readout. So that's one possibility. The other possibility would be that that if you had more of the sensors, you might have like a higher readout. But now I'm maybe it's not correct because I'm thinking that that you would still have like the ATP to ADP ratio. Like I guess if the sensor did not itself account for the ratio, like it's one sensor giving you these two values, ATP to ADP. And instead it was a sensor only giving you like ATP information, then that would be problematic. But in this case, because it is a ratio, maybe it's not problematic. Yeah.
Okay, so what do you think would be a good choice here?
I guess the D.
Uh-huh. And yeah, because um yeah, so sure, we know that fluorescence intensity is proportional to the number of sensor molecules excited. Um, and we wanted to see if that is indeed the case, or is that something that we just see an increase in because of higher expression of Y32? So, um, so yeah, the ratio part is what makes it um like take into consideration the number of the Y32. So yeah, so that should be your answer. Um
Yep, I agree.
yeah.
It's funny how I was like off because I think I was thinking of that thing we wrote down of the proportionality of
Mhm.
numbers excited. Yes.
Mhm. Yeah. So like, we don't know if we get something like this, we don't know if it's like, let's say, like 70% of the um Y32 that gets that fluoresces at this wavelength, or if it's just like a smaller percent, but just a higher number of the molecules. So, but yeah, the spectral ratio intrinsically normalizes for the amount of the biosensor, and that means that it's that means that we already have taken into account the uh possibility of there just simply being more Y32 when we do the ratio. So yep,
Yep.
that would be that one. Yeah.
My screen is frozen. Let me
Oh,
just reload it. No?
oh, maybe you still have the annotate thing on.
Oh yeah, I think so. I've never used this annotated.
Mhm. Yeah, there should be like a on the toolbar like a mouse cursor icon. That oh yeah, you got it working. Okay.
Yeah.
Oh, so here's exactly what we talked about.
Yeah.
Fluorescence. Yep.
Um, in a follow-up study, the emission spectra of both bound states of the biosensor were shifted to longer wavelengths than the excitation spectra shown in figure 2. Shit. Emission spectra, not excitation.
Okay.
What is the most likely cause of this shift? Okay. The emission intensity peak is usually lower than the excitation peak. There is a loss of vibrational energy when electrons move from the excited to the ground state. The excited at a wavelength at the peak of the excitation curve. I think B. I don't know if I would say that. Is it vibrational energy? I don't know. I guess I could go through like what I'm thinking. So it's asking me like why in another experiment, and now they're looking at emission, not excitation. Um they were looking at longer wavelengths instead of here than here. And I'm thinking it's because like if you're emitting, you've already like, as we like discussed earlier, you've like lost a lot of energy, like shifting it to another energy level. And like there's less energy, and like that's why you would be looking at lower energy, lower wavelength, longer wavelengths, um, when the electron is emitted. So that's why I would go with B. I may be wrong, but that's where I would go.
Yeah, no, that's good. This is exactly what when I talked about, you know, oops, when I talked about like getting excited up here and then it can relax down without emitting a photon and then it can emit a photon. So we know that in fluorescence, the energy of the photon absorbed is larger than the energy of the photon emitted.
Yes.
So in other words, that means that the wavelength um, of excitation is shorter than the wavelengths for emission. And they're telling us that, they did this and they looked at the emission spectra, which is different from the excitation spectra, and they found that it was all shifted to the longer wavelengths, which is what happens in fluorescence. So then just asking what's the cause of this shift. So we're looking essentially for either something talking about resonance stabilization, conjugation, all that stuff. And B describes it as a loss of vibrational energy when electrons move from the excited to the ground state. So that can be something that would, you know, uh, cause a lower energy level to be reached in fluorescence. So I would go with that. I would go with B as well. A says the emission intensity peak is usually lower than the excitation peak. What are your thoughts on that?
I think peak is usually like referring to like the peaks here. Uh, and that's more like intensity maybe, or like fluoresce like the which I think the question is asking like why the wavelengths were shifted, not the height of the curve. Yeah.
Good, good. Yeah. We can't say that, that the intensity peaks are different because that's not really something that we're able to measure. Um and they don't tell us that that happens. They just tell us that it gets shifted to longer wavelengths. So that's out. Then C says fluorophore is typically excited at a wavelength at the peak of the excitation curve. And so that's something that can um what are your thoughts on that?
Excited at a wavelength at the peak of the excitation curve. Mm. Um, I just feel like it's not answering the question, but
Exactly. Like you'll see this a lot. You'll see true statements that so C is a true statement, but it doesn't answer the question. The question asks, what is the cause of this shift in emission spectra to longer wavelengths? Doesn't answer that. D says detection was selected at the peak of the emission curve. Again, that doesn't explain why there's a shift to longer wavelengths when you're looking at emission spectra. So yeah, this is exactly it. And so they're talking about it as a loss of vibrational energy, but they can say a lot of things here, like conjugation, blah blah blah. So this just talks about. So this is just something that is really high yield because they'll ask you a lot about fluorescence.
I see.
But yep, you're right.
Okay.
Mhm.
Let me just write something down.
Mhm.
Okay. When Y32 is expressed within a normal cell, What is true of its nucleotide binding site? Oh, there's more ATP. We talked about it here. Um, oh, D. I think D.
Let's see.
Because yeah.
So okay, tell me a bit about it.
Um, so yeah, at first I thought it would be something like which one is more present and like what is it more likely to be like occupied by like that which has like a higher concentration, but like it would be like ATP in this case if it has like because ATP is, if I remember right, more the higher concentrated within a normal cell. Here, it's of a normal cell. But since that's not an option, I thought it would be like it would be always occupied by this or that. Um,
Mm-hmm.
yeah.
Yeah, yeah. I guess I don't like the word always, but I think that would fit here.
Yeah, it sort of gives the impression that it's always active, like it always has something bound to it.
Mm-hmm. Yeah, I would think so. B and C are out, and then honestly, it would either be A or D. Like A, if it says it's most likely to be occupied by ADP, I think we said something about how the ADP concentration was.
Lower.
Oh, it was lower. Okay.
Yeah.
Um,
Wait, well, I don't know. Like we said, because it's like hundreds of micromolar versus 1 millimolar.
mhm,
And one millimolar was higher for sure.
mhm.
I mean, I guess this part makes it a bit like I would have to like normally write it down because it's like I don't know, like I don't trust myself with math.
Uh-huh.
But I think it would still be like 1 millimolar would be higher.
Mhm. Okay. So yeah, we can do that. And yeah, is there any more?
Yes. Y32 is pH sensitive. This can be a problem when employing it to monitor changes in the energy balance of a cell over time because um C. So I feel like it wouldn't be A, and my logic here is because it's of a cell, so like you're only comparing one cell. That's why I'm not inclined to choose A.
Okay.
Um, so B, it's the ATP to ADP ratio of a cell depends on pH. So this is sort of implying that like whether you're like your metabolic states or your like your if yeah, I just feel like it's wrong. I feel like if anything, it would be in the other direction. Like your energy state would affect pH and yeah. So like ATP is affecting or energy, ATP, and then pH.
Mhm.
So I feel like the directionality is wrong. And then D, let me see. Decreased pH favors a CPVB state. Um, Decreased pH, you would have higher proton, so it would favor the A state. So I feel like if you have decreased pH, you would be more likely to be protonated, so you would probably be in the A state. Is that correct? So I think like C is the only one left, and I feel like it makes sense.
Mm-hmm. Yep. Very good. Yeah, I think that was all of it.
Yeah.
So yeah, how do you feel about how we went over it? Like, how do you feel about it now?
I feel better. Um I think this like wavelength and spectra, that could be a little bit confusing sometimes. Um, um, yeah, I think that it wasn't too bad of a passage, but I did terribly on it. I think I only got one question right when I did it when I did the test
Mhm.
or
Do you
two.
find like the way I wrote these proportionalities to be helpful?
Yes. Yes, I did.
Okay.
I also feel like when I so how do you do it for like Chem Phys? Would you always recommend reading the passage and then the questions? Because I feel like it's working well for me for like the other sections, and maybe because I can do them faster. But for these, I tend to like I'm reading it and then I forget what I just read
Hmm.
because it's very loaded.
Yeah. What, so you're asking about CP? I forgot. Is this CP section?
Yeah, CP.
Okay. So you're asking like if you should like read the passage first or
Yeah.
yeah, so I would say that for the most part, yeah, you want to do that. If you're running out of time for CP, you that's the only section where you can perhaps get away with reading the questions first because a lot of the questions are kind of like pseudo discretes. Um, so yeah, that could be something that you can do if you are running out of time. Um, but only if you're running out of time, I would say to do that. But the other thing you said about like, you feel like when you read something, you like forget it right after. That's where writing out proportionalities and things like that can be really helpful. Um, so that's why when we talked about this, like for instance, when we talked about it can sense the ATP to ADP ratio, but we don't know exactly like when they first mention it in the first sentence if it's proportional to that or not. But then they tell us that there's a sensing domain and a reporting domain, and they say that if Mg2 plus ATP binding like happens, that causes a conformational change in the sensing domain, which then gets transduced into a conformational change in the reporting domain. So that's why I wrote like these proportionalities here.
Yeah.
And so that way I know that if something increases, these other things increase, and if something decreases, these other things decrease. But that can help you, like, nail down something concrete for like that paragraph. And I would just say to continue doing that. We can do like another one to show how, it can be adjusted. But, and when we got to paragraph 2, we had some information, but I feel like the information wasn't so important that we wrote something about it. Um, but we just got, the information about, I guess, the concentration of these things and high affinity and stuff like that. And then when we see the figure 1, we see what we should be expecting, which is them asking about fluorescence. And so, we see the two. Well, first of all, we see lots of conjugation and all that stuff, so expect to see some fluorescence questions. And we just have like sure an A state and the B state, and they tell us that the A state is favored in the ADP bound conformation, and the B state is favored in the ATP bound one. So I drew that here. I wrote A state proportional to ADP bound, B state proportional to ATP bound, and then they talk about in the last paragraph, they talk about the um the ratio. So if you go down to the last paragraph, they talk about the ratio, spectral ratio, right, of the peaks is directly proportional to the ATP to ADP ratio. So that's where I wrote fluorescence intensity of B at 500 nanometers, that's the peak there, divided by the fluorescence intensity of A at 425 nanometers, which is the peak for A. That's the spectral ratio of the peaks, right? And then that's directly proportional to the ATP to ADP ratio. So pretty much for almost every paragraph here, we read what they were telling us and we wrote out proportionalities, and that makes it so that it's, we can make sense of it.
Yeah, it helps.
Yep. And then yeah, just a lot of questions seem to be about fluorescence, so it was good that we went over that.
Yep. Yeah, it's a good passage. All right. So I guess I do have like it was passage 3 and also didn't do pretty well on these discretes.
Oh,
So
that one seemed to be about radiation
this,
or active decay.
oh, we talked about this. This is the one I got right and not the others.
Uh-huh.
I think I ended up choosing a gamma particle. I remember, I think.
Uh-huh.
Just because we discussed how it has no mass. Um I was a bit, I was thinking when I was going over this of like a beta particle as well, because it also doesn't have mass. We discussed, I think. But I think it yeah, I feel like it would still have a charge, and like I think we talked about like beta minus and beta positive, and wasn't one a positron?
So this is where that negligible thing comes in, that is kind of iffy, like sometimes, like for instance, so okay, we know alpha particles out, it's the one that it's the only one that, well, it has the most mass, right? And the beta particle, can be like an electron or a positron. So both of these we have, like, or we're taught have no mass, right? So it's kind of hard to answer this then, because beta particle and positrons we know to have no mass, as does a photon, like a gamma particle. So this is where, what I talked about how if something is negligible, it doesn't necessarily mean that it has zero mass. Like the mass of electrons and stuff are just 1/1800th of the mass of a proton. But so that's why this one, the answer is going to be the gamma particle, because the gamma particle, a photon of light, has no mass, whereas a beta particle or a positron or an electron we consider to be having negligible mass, but that still counts as mass. But it can be annoying, you know, it can be pretty annoying because sometimes they can, like that question that we did, the thermodynamics question that talked about like open closed systems, isolated systems. Like, I wanted to choose the answer that involved I think like mass exchange, just because it says mass is negligible. But it was the correct answer was with consistent with saying that the mass being negligible is zero mass.
Yeah.
That's usually the case, but then for this question, then their negligible is not equal to zero, right?
Double standards. Yeah.
Yeah. So yeah, this could have been tough, but yeah, you chose the right answer, yeah.
Yeah. I guess like I'm open to doing these, or we could go over the passage, because like I did, like, of the whole test, like this is a part that I did worse on, like these two passages and this set of discrete. I don't know why, like, oh, like it was like a rough, like bad stretch. Yeah, so
Got it.
yeah, I feel like we do a lot like when we do the passages, so we might do that instead.
Yeah, and this one talks about radioactive decay, so this is going to be very like relevant to what we talked about last time.
Yeah, yeah.
So
Okay.
yeah,
Yeah, so let's do it. So gamma knife surgery. Let me put our images somewhere that it's not annoying to me.
there's still. I thought I cleared all
No, it's like the videos. Um,
videos.
um, it's good now. Gamma knife surgery offers benefits over traditional neurosurgery that make it a popular choice for the treatment of certain neurological malignancies and disorders. Um, okay, so this GKS is better than TN. Uh, and I guess I could write that down, but it's probably not that important.
Yeah,
Uh,
I would say not very important because they're probably, not gonna really talk about how it's a benefit. They're just gonna talk about the gamma knife surgery part,
yeah.
likely. So yeah.
No traditional knives are involved in gamma knife surgery. Multiple beams of beta and gamma radiation are directed towards an area of the brain without a physical incision being made. Okay. You're using like energy basically to radiation for the surgery.
Hmm.
Because of no direct physical damage is inflicted, GKS has lower risks of infection, leakage of CSF, and hemorrhage than TN.
So
Okay.
so tell me what they're telling us here.
So gamma knife surgery, it's using beta and gamma radiation to do the surgery. And so because of this, like there's less side effects. Yeah.
So there's no physical incision, there's no direct physical damage, right?
Yeah.
So so yeah, it's better for these reasons. Okay, cool. Continue.
The ionizing radiation used in gamma knife surgery is produced by the decay of radioactive isotopes in a device around the head.
Okay. What can you tell me about ionizing radiation?
Um, Ionizing, so you're exciting electrons. Uh,
Are you just exciting them?
mm, um,
Because think about if it causes ionization, right? That means that you're like removing the electrons, right?
oh, I see. Yeah.
Completely. So in order for something to become an ion. So so yeah, ionizing radiation is just a type of high energy radiation or light that is capable of turning, stripping electrons off of things.
Okay.
Turning things into ions, yeah.
Um,
Mhm,
this is produced by the decay of radioactive isotopes.
mhm.
So
Yep.
yeah, radioactive isotopes, sort of, I think we talked last time about like carbon dating and like carbon 14. But here it says the decay of these isotopes.
Mhm.
Um,
Yeah.
let's see. So is it like these isotopes like changing and like what is it? Like going through like beta minus, beta plus, like all of these things. Um, and so there's a change in it, and this is causing the ionizing radiation,
Mhm.
maybe. And in a device around the head. Cobalt 60 is commonly used for this purpose. It undergoes, yeah, it undergoes beta minus decay and has a half-life of 5 years. Okay, I guess I could highlight this in beta minus.
Yeah. So now we have um some information about one of the radioactive isotopes that could be used for this, and they're talking about cobalt-60, and it undergoes beta minus decay, so we know what type of decay it undergoes. And they give us the half-life of 5 years, so they'll likely ask us stuff like that,
Yeah.
about that stuff. Okay, good.
Hundreds of beams of radiation emitted from molecules containing cobalt-60 are focused on a specific location, like layers are focused. While individually, each beam has very little effect on the brain tissue it passes through, a strong dose of radiation is delivered at their convergence point.
Okay.
The radiation is strong enough to cause molecular electronic transitions by exciting electrons to higher energy levels in molecular orbitals.
Mhm. So okay, so um they take molecules containing the cobalt, and you know that's going to be releasing all of this um beams of radiation, like for beta minus decay and stuff, and they focus it all on, or converge it all on, like a certain point, which will make that radiation strong enough to cause electrons to go up to higher energy levels.
Yeah.
And
Yeah.
yeah,
The okay, I think this is right. In some molecules, a beam passes through. The radiation is sufficiently strong to produce molecules with unpaired valence electrons by way of electron ejection.
all right, what's that talking about?
Um, it's it's what we just like if you go past like the valence shell of energy, and you have enough energy, like you may eject it. Um,
So that's the photoelectric, well, the work function thing, right?
right, it's more than the work function, so you would have that extra.
Good.
However, the radiation is only intense enough to induce cell death at the convergence point. They mentioned the convergence point. The machine is precision calibrated, so this point is targeted at a malignant growth. Okay, I see. So they're shining a lot of these, what is it? I guess cobalt-60 beams of radiation lasers onto a tumor, and like one wouldn't do anything, but when they have a bunch, they have a higher intensity, they're able to like target it at like probably cancer growth, and that's the surgery.
Yeah, so it can, if you make the convergence point at a malignant growth, it can cause cell death there without causing without being like as invasive as, like, if you use like a scalpel or something.
Yeah.
Oh, okay, awesome. Good, good, good. Oh, and I liked how you noticed that they're talking about intensity when they talk about more of the beams at converging at a point. So okay, great. All right, continue.
Cobalt-60 is not the only isotope that can be used in GKS. Researchers performed a clinical trial comparing the effectiveness of GKS with cobalt-60 to GKS with isotope X. And for treating this pituitary adenomas, X undergoes beta plus decay, that's different, and has a half-life of 12 months. In order to measure the relative efficacy of this, they assess relative tumor volume 1 week, 3 months, and 6 months after the surgery. Additionally, they reported the rate of unintended neurological side effects 6 months post-surgery. The results are shown below in table 1.
Okay.
Okay, table 1. So cobalt 60 and X. 1 week post-surgery, it looks like cobalt is better. 3 months, it looks like X is better. 6 months, X is better, but X has more unintended side effects.
Mm-hmm. Perfect. Good.
Okay.
So we could also say perhaps that beta plus decay, which is positron emission, seems to be better 3 months after surgery and 6 months after surgery, but can also cause more of these side effects.
Yeah.
Okay, cool. So doesn't seem like we needed to really write much for this, but does it kind of make sense what they're talking about here?
Yeah, it makes sense.
Okay, good. So, um, okay, awesome. So let's try. Oh, this is question 13.
Oh, better. Yeah, better. The knives used in GKS can, 1, generate free radicals, 2, excite electrons to higher energy levels, 3, eject electrons from molecular orbitals.
Okay.
Um, the radiation is strong enough to cause molecular electronic transitions by exciting electrons to higher energy levels in molecular orbitals. So 2 is correct. That doesn't help us much, except probably A is not correct. Eject electrons from molecular orbitals. The radiation is sufficiently strong to produce molecules with unpaired valence electrons by the, okay, so it can eject electrons from molecular orbitals, so 2 and 3 are correct. Do cell death at the convergence point, okay. I think I feel like 2 and 3 are correct. I don't know about 1. The passage doesn't seem to, I didn't see where it said free radicals. So my, like, right now I would go with 2 and 3, but like I don't know about 1.
You said 2 and 3, you don't know about 1. Tell me
Um,
about
so
1.
so 1 is generate free radicals. Um,
Tell me what that is.
it's like when you have like an electron, or can you have like an extra electron or like an unpaired electron, on its own, so it's very high in energy. Maybe I'm not too sure. I can recognize it drawn, but I don't really know exactly what it means.
All right. Do we so what did they mention in the passage about this stuff?
About free radicals?
Yeah. Did they maybe not by by name, but did they say anything about that?
Well, they mentioned side effects, and I feel like free radicals would lead to like ROS or like side effects. Um, but let me see. Molecules with unpaired valence electrons. Oh, I see. Yes. I see. So like when I read that, I just thought they were referring to 3 eject electrons from molecular orbitals.
Mm-hmm.
So they're doing 3, but they're like they are rejecting it, but when they eject it, they're also generating free radicals, it seems. Yeah, so I guess it is 1, 2, and 3. But can you tell me more about free radicals?
Mm-hmm. Yeah, no, it's pretty much what you expect, which is an unpaired. So like electrons, they must be in pairs because that's how they're stable. So if they're not in a pair, they are called free radicals, which means that they're super, super unstable. And if it's super, super unstable, it means that it's super, super reactive. And that's going to cause damage. And that's why, like, to fix it, you can have like, well, that's also why it can be called like reactive oxygen species. Um, and that's also why to prevent it, you can, you know, take something like an antioxidant of some sort. But yeah, essentially that's what it is. A free radical will be an unpaired like valence electron. Like, if we had like two bromines attached, uh, and you add like HV, which is like energy in the form of like light, perhaps, um, it can cause like that, which would be like bromine, like two bromine free radicals. You know, this is like initiation, and then there's like it's not elongation, but it's something like that, and then it's like termination. Do you know what I mean?
Yeah. What do you mean by initiation, elongation, and termination?
Oh yeah, so the initiation is the creation of um the free radical, and the other one is called, oh, it's called propagation.
Okay.
And that just makes more of that free radical. And the last step is termination, um where I'm trying to find a picture here. Okay, so yeah, so initiation is the step where the free radical forms. So that's this step over here. Propagation would be continuing to make more free radicals uh in different molecules. So let's say that we had like methane or something, like CH4 plus the bromine free radical. The bromine free radical is going to want to not be a free radical essentially, so it can take a proton from the methane to give you and to leave that methane as a free radical, so this can turn into CH3 plus HBr. So that will cause propagation or the creation of more and more free radicals. And then the last step would be like, if you had like whatever, two bromines again, like free radicals that bind together to turn into that again, the two bromines. So that's like the general mechanism for free radicals, the creation of free radicals with the initiation, the spread of more free radicals through propagation, and then the termination or ending of the free radicals, turning back into the original molecule. So, yeah, so that can be something that happens. Um, but aside from that, yeah, you pretty much understand what free radicals are, and I think that it's just in this paragraph here that talks about free radicals. So I think that's what would lead us to choose um, Roman numeral 1 as being something that can happen.
Yeah, I see this.
Yeah. Like we know Roman numeral 2 happens because they tell us about exciting electrons to higher energy levels here.
Yeah.
And ejection of the electrons we talk about here as well. So And then yeah, the free radical thing we also talk about in between.
I see.
So yep.
Okay, a hospital purchases brand new GKS Co and X machines. 5 years after installation, what is the expected ratio of the total atomic mass of material in the CO machine to that in the X machine, assuming both machines start with the same mass of radioactive material? This is a half-life question. Okay, so they want the ratio from CO to X, and so it says CO is 5 years, and this one is 12 months. So I have to, I guess, half-life is usually in years. I think it's in years, but I don't know. I'll assume it's in years. So I would say 5 years to 1 year. And so 5 years after installation, I should have lost a lot more of my X than of my CO. So I expect it to be in that direction. So I expect it to not be C and not be D, because I expect to have more CO than X.
So let's make sure we understand what the question's asking.
Yeah, okay.
So what is it asking?
So it wants me to say how much of each material I will have at the end of 5 years. Assuming I had the same initial thing, the ratio.
What is it asking though about the ratio?
The total atomic mass of material.
Mm-hmm.
Total mass.
Yep.
But isn't that the same as like amount of material?
Is it?
Do they even
So what type of decay do
Oh, wait. Sorry, could you finish your question?
Mhm. What type of decay does cobalt and X undergo?
So cobalt on X does beta plus.
Okay. Um, well, X does beta plus, right?
Yes, X does beta plus, cobalt does beta minus. Sorry.
All right. So do those particles, beta minus or beta plus, have any mass?
Do you mean like what's emitted?
Oh, sorry. Like the beta minus particle, beta plus particle, do they have mass?
No.
Okay. So as they undergo decay, should the mass change?
I see.
Mhm.
I see. It shouldn't change if they don't have mass because they're not losing mass.
Yeah. So this is a super like trick question. Um, because that's why I was asking you, like, let's really make sure we know what the question stem is asking us. And it's very easy to overlook this. It's very easy to overlook this because we are already thinking about when we read the passage about, okay, cobalt half-life 5 years, X half-life 1 year. So a 5-to-1 or a 1-to-5 ratio would be very, logical, right? Um, but yeah, we have to be very careful because they mention just the total atomic mass, and that doesn't change because, you know, the beta particles don't have mass. We don't really consider them to have mass, so the total mass of the material should not change, right?
See.
So super trick
Yeah.
question here.
So when they use half-life in the passage,
Mhm.
what are they referring to? Like, is it just like the decay, but not like
It's
just the rate of the decay? That's what they're referring to. Okay.
yeah, no. So like so for instance, like if you had this big thing of cobalt 60 and 5 years pass, then you would have half of the amount of cobalt 60, and the other half would be whatever cobalt 60 uh decays into, right? So whatever that would be, something over here. But yeah, it would be 50-50 after. So a half-life is the time it takes for half of something to decay.
I see. But is it like it's not changing because it's just say of material in the cobalt machine rather than like of carbon cobalt 60 material? Like if they had specified, then that would matter.
Mm-hmm. Yeah. So if they said something like, what is the expected ratio of the cobalt, um, you know, uh, not the mass part, but if it just said like what is the expected ratio of the material in the cobalt machine to that of the X machine, right? That would then be something along the lines of this. But since they're just saying the expected ratio of the mass, that won't change.
Okay.
So, um, so yeah, like for instance, like 5 years, right? We should have uh half of the amount of cobalt 60, right? But X, right? So like after 1 year, it'll be 50 or 0.5 X, right? And then after another year, it'll be 0.25 X. Another year, 0.125 X. Another year, 0.0675 X. And then 1, 2, 3, 4, 5, the last year will be half of that. So that's like 0.33 something, right? So if they asked us what's the expected ratio of, you know, cobalt compared to X, right? Um, after 5 years, you'll have 0.5 or half of the cobalt 60 and like 3.3%, you know, uh of the X. And um so yeah, that would be I guess like 0.5 over like 0.033, something like that, or like essentially just how many times. Well, I guess I could also do 1/2 for the 0.5 and then dividing it by 0.033. That's so 1/3 is 0.33. 1/30 would be 0.033, right? So if I'm gonna multiply it by its reciprocal. So um So like it'll be times the 30 part, which will just be, well, 2 goes into 30 15 times, so something like whatever
Yeah,
in here. So I guess that would be kind of close to the 16:1 ratio.
I see.
So yeah, so that would be a more normal type of half-life question.
I see.
But um yeah, yeah.
I see. I get it. Difficult, tricky.
Yeah, super tricky because of that total atomic mass part. I'm sure like almost everyone probably got this wrong.
Yep. That's the answer. Um, which of the following best explains the tumor reduction capacities of the isotopes observed in the study? Okay, so this.
Yeah.
GKS X emits more radiation per unit time, causing more self-death. Um, GKS X emits narrower radiation streams and target the tumor. I don't think so. The cobalt 60 isotope in GKS cobalt has a longer half-life than X in GKSX. It does, but let's see. And GKS cobalt does not emit sufficient radiation to kill tumor cells. But it seems to do. The tumor reduction capacities is what we're looking at. So I feel like I don't think it would be D, just because like it is reducing it. So like in my mind mentally I would like disregard that one, like strike through. For A, I'll still leave it in the running, even though I don't get any information about like intensity, or do I? But I do know that it does cause more like there's more unintended consequences, like side effects. And it does cause a lot of cell death, so I'll just keep it there.
When you said intensity,
We said intensity?
um, did you say intensity
Yes.
for
Yes.
where?
For A.
So show me where there's intensity.
So I was just thinking of radiation per time. So
Yeah.
like, yeah,
Intensity is like power or watts over square over area.
I see. Um,
Mhm.
Here it's energy over time.
Um, here.
Yeah.
So yeah, so here we can say that that's just radiation per unit time. Um, so what do you think that would be?
Power.
Oh, well, so we don't really know what they specifically mean when they say radiation, right? For us to say that it's power, but we can at least say that. So for instance, cobalt 60, right? Releases a beta minus, well, releases a beta minus particle after 5 years, right? So the radiation that it emits is talking about like radioactive decay. So it's basically a beta particle every 5 years, right? So like 1/5. Whereas X in just 1 year will release a beta plus particle.
Yeah.
So that would be one like radiation or radioactive, particle per 1 year, right? So this is like one radiation per unit time, and then this is like 0.2 radiation per unit time, right?
Yeah. So it's a true statement.
Uh-huh.
Yeah.
So, yep. So, okay. So, yeah, we want to explain why tumor reduction it seems to be enhanced when we use X. And yeah, X seems like it does emit more radiation per unit time.
Yeah.
So that could be something that can cause the cell death.
Yeah. And it does cause more self-death. So.
Yep. So, okay. What are your thoughts on B?
I know it has more unintended side effects. So I don't think it would be narrow radiation. It would probably be wider radiation.
Uh-huh. So, um, let's see. Okay. Um, I guess thinking of narrower radiation streams as being higher like intensity because intensity is inversely proportional to area. Um, so, but yeah, we don't have anything that in the passage that suggests that, you know, the GKSX emits narrower radiation streams, right? We don't really, we never mentioned anything like that, right?
Yep.
Okay, and then C.
It does have the cobalt 60 isotope has a longer half-life than X, which it does,
Mhm.
but it's sort of what you explained here. So it's kind of like A or C. They're like they're flipped.
Mhm.
So I guess yeah, the way that you explained A would seem that if you're emitting like less B minus, which is what's causing like the tumor reduction, that you're not improving your tumor reduction capacities. You're reducing your tumor reduction capacities.
Yeah,
But
so if we wanted to do like radiation per time, that would be an inverse proportion to half-life, right? If you have a if you have a shorter half-life, you should be producing more radiation per unit time compared to if you had a longer half-life, right? So yes, A talks about radiation per unit time for X, which would be the same thing as saying like this is because X has a shorter half-life. So C is wrong because the longer half-life um, of cobalt um, makes it less makes it work less effectively at the tumor size.
yeah.
And then what about D?
Um, um, well, it does kill tumor cells because like we can see the tumor
Yep.
cell size going down.
Yep. Good. So okay. So D is directly contradicted by the table. C would give us the opposite type of results. Um, B, we don't have anything that suggests that. And then A is what would make the most sense. And I just want you to be able to see like how a passage can introduce some new type of, I guess, I don't want to say unit, but like this is its own thing that's not the same thing as power or intensity.
Yeah.
Um, but we can just take that to mean like if something undergoes radioactive decay, it gives off that decay product. So the radiation could be equivalent to that radioactive decay product. So, so X emits more radiation per unit time because in 5 years, cobalt-60 will emit just one radiation thing, like, but in 5 years, X will emit 5 times as much of the radioactive particles. So, yeah. So this.
Different way of viewing the whole half-life, because it's usually not in this context that I've seen half-life.
Yeah. So think about the inverse proportion with half-life and radiation per unit time.
Yeah.
Because also, like, if the half-life of something is short, it's more dangerous in terms of radioactivity, right? Because it's just shooting out these radioactive,
Yep.
particles and stuff. So, yep.
All right. Another question. Free radicals from ionizing radiation are highly unstable and have carcinogenic effects. These effects most likely result from damage to I feel like whenever I think carcinogenic or carcinogenic, I think that it's something to do with like probably D, nucleic acids, but I don't know. Just because I'm thinking of so like I'm thinking of of like mitosis and like excessive like cell cycle problems. So I would think of nucleic acids. I know Golgi apparatus is like packaging and like distributing elsewhere. Phospholipids, I guess I'm thinking of like the membrane, like phospholipid membrane and like entering or exiting the cell, mostly, and then proteins. Yeah, I think I would go with D, but yeah. What do you think?
So tell me why you would go with D.
Because I feel like if it's like cancer producing, like I'm thinking of like P53 and like mitosis and like just basic like transcription part and everything else I feel like would not be as directly involved in like being cancerous.
Yeah, the ionizing radiation can, if it damages the protein, sure, it'll damage the protein of that particular protein. Um, Phospholipids, it would damage like the phospholipids of like the cell. Golgi damage that part of the cell, but keyword here, which is what you highlighted as carcinogenic, which means that it's cancer causing. So the ionizing radiation, instead of it just destroying what it touches, what can it destroy that would make it so that it can lead to cancer from, not having to be directly related to what the ionizing radiation would damage? And yeah, that would be DNA, because that would be something that would cause cancer. So, good job.
Yep. All right, final question of this passage.
Mhm.
Which of the following types of electromagnetic radiation would have the shortest wavelength? Yeah, I was clueless when it got to this question.
Okay.
Mostly because I'll try to think. So I know that it's higher energy, like which one am I ejecting? Would have which of the following types of would have that shortest wavelength, like highest energy? Ejects an electron from an sp orbital. I guess that would be closer to the nucleus,
Okay.
right? I don't know. I'm just thinking out loud. Maybe that's wrong. From an sp2 orbital, from an sp3 orbital, radiation that excites but does not eject an electron from an sp3 orbital. I feel like it has to be ejection because like you would have like it takes less energy to excite and more energy to eject. So I feel like it wouldn't be D. Now this is a whole hybridization thing. SP, SP2, or SP3. And I guess I could think of it like, well, we talked about the different shapes. Um, I don't know how that helps me though. Um,
So
and when I was going through this, I feel like now I'm more thinking of it like, where is it located within, like, is it closer to the like nucleus, and like would that take more energy or less energy or farther away? But I don't know. Yeah.
so yeah, let's think about um the shapes and how that can contribute to the type of bonds that are made. So for instance, um
Oh, is this single bond, double bond, triple bond?
yeah, something like that. Do
Uh,
respond to which?
SP would be SP3 would be single, SP2 double, SP triple.
Yep. And um yeah, go from there. So then the highest or the strongest bond would be like the triple bond, right? And if
Mhm.
it's a stronger bond, it's a shorter bond, right?
Yes.
So
It would have its electrons closer.
mhm.
Um, so so you would need like more energy in order to like eject that electron. That's what
Yep.
would have the shortest wavelength. So I feel like, I guess the question, which of the following types of radiation would have the shortest wavelength? Are they referring to like what I'm shining at, like what light, or like what is that what they're asking? Like, which of these would I need the highest energy tool in order to do this, in order to like eject something? Is that the question?
Yeah, so I guess like if they're asking you like okay, so you got rid of D, so we're looking at ejection. So
I think it's A. Yeah, I think it's A.
yeah, because this is talking about um like so shortest wavelength would mean highest energy. So radiation that ejects an electron from which type of orbital, which of them, like which one would produce the most highest energy, like photons, right? Or require the most highest energy for photons, and the sp orbital is the strongest bond, which is the shorter bond as well. And uh so to break that up, or to cause, an electron to be ejected from that orbital will require the most energy.
Yeah.
But
I-
yeah, essentially what you said with triple bond, double bond, single bond. So that.
Oh, I see it. I see it now. I wish I could have seen it before.
Yep, yep. Yeah, and also like I guess like I should say like the percent of S would be proportional to or inversely proportional to distance from nucleus. So like SP has 50% S, whereas like SP2 would be like, I guess like 33% S. And then SP3, I think would be like 25% S, something like that.
Yep.
So more S character means, closer to the nucleus. So that can also help.
Yeah, it does.
Mhm.
Good.
Yep.
Um, could we go over these discretes?
Yep. So
Yeah. Um, Yeah, well, I guess this is what, like, I feel like I waste a lot of time with math. So I just like ended up like probably skipping a lot of like the math things. Um, here it's just only two. Um, yeah, so I guess it's worth like going over them.
Mhm.
Person pushes horizontally on a 50-kilogram crate, causing it to accelerate from rest and slide across the surface. If the push causes the crate to accelerate at 2 meters per second squared, what is the velocity of the crate after the person has pushed the crate a distance of 6 meters, assuming there is no friction? So I have the mass of the crate, I have the acceleration of the crate, and I have the distance, and they want me to find the velocity.
Do we have an initial velocity?
No, it's 0.
Yeah, yeah. So we do have initial velocity, right? It's 0.
Yeah. Yeah. Contradicted myself there. Yeah. Um,
Well,
Sometimes I feel like there are questions in which I have to apply like the like kinematic equations, and then there are others in which it's just like a simple like acceleration, and like they wants me to find velocity, and I'm giving the distance. Um
mhm.
I guess I could start with like acceleration is velocity over time, right?
So what you just said about kinematics, right?
Yeah. So I feel like there are problems in which I have to use like the kinematic equations,
Mhm.
versus there's others that are could be like simple, like like using like the basic formulas. Um,
So
but I am giving it an initial velocity of 0, so it could be that it is like one of the kinematics.
yeah, so what you did um first is what you should do. You take an inventory of the information that they give us. So we have a mass, we have a initial velocity of 0, we have an acceleration of 2, we have a distance of 6, and we need to find the final velocity. So um so writing it out like this can help us determine what. So we're going to be using kinematics, right? Because we have, all these different, you know,
Um, could
val-
we use the equation that's like x equals like the average velocity times time? Would that be useful?
uh,
Uh, do I have a time? No, I don't have a time. Don't worry. Not useful. Um,
yeah. So why don't you tell me what the kinematics equations are?
yeah. Let me just take a second to dig them up for my brain. So there's like velocity final equals um velocity initial times time. Is that correct? Plus, no, sorry, velocity initial plus AT squared. Yeah. There's X,
So let's think about this one because you said AT squared. So like,
just AT. Yeah.
because yeah, if you subtract V initial on both sides, you would get delta V is equal to AT, and then you divide T on both sides, you would just get, accelerate like the equation for acceleration like this.
I see. Yeah.
So okay, so this is one type. All right. What's next?
Um, there is X equals velocity initial times time, I think here. Um, plus AT squared over 2. Um, there's velocity squared equals velocity initial squared? Yeah. Well, yeah. Plus um, um 2ax. 2 times a times x. Yeah. I think
Good,
so.
good,
Mhm.
good. Okay, awesome. So, which one do you think we should use?
Um, the last one.
Yeah. Why?
Because I have like the acceleration, I have the distance, and I know that my velocity initial is 0. So that would give me my final velocity, which is what I'm at.
Because we don't have time, right?
Yeah.
Good. And, you know, you also probably recognize that the mass here isn't going to be relevant. Um, why wouldn't it be relevant?
Um, I'm only doing horizontal movement, not vertical movement.
So if we had friction, then the mass would play a role, right?
I see.
Because like the force of friction is, the coefficient of friction times the normal force, right? And the normal force is typically equal to the gravitational force, which would be the mg.
I see.
So if this, if this, so they say it's no friction, but if there was friction, then the mass would play a role.
I
But
see.
since there isn't friction, the mass won't play a role. So yeah, we can just now plug it into this over here to solve for the VF. So let's do that. So this is going to be equal to 0 plus 2 times 2 times 6, right?
Mhm.
So,
So
yep.
24
Mm-hmm.
square root.
Yep.
Um, um, I guess I could use the options here. And
So
I probably be 4.9.
yeah, so how would you do something like this?
I guess I could basic math that I'm forgetting. I could break it up into like its component ones that I know. So like I could do 4 times, wait, no, 16 times something. No, I don't know. Yeah, I guess, I don't know. This is a long time ago. I could like find like roots that I could find. I could break up 24. That's what I'm gonna say.
Yeah, yeah. So you can even compare it to like, okay, the nearest like perfect squares. So like
Yeah,
radical 25 would be 5, and then radical 9 would be 3.
I
Right.
see.
And then so let's see which one would it be closer to, right? So 24 and 25 are they're separated by 1. Whereas 9 and 24 are separated by, I guess, like what, 15.
Yeah.
So it's going to be it. So it will have to be between 3 and 5. So A and B are out, but it's significantly closer to the 5 than it is to the 3,
Yeah.
right? So D should be our answer.
Yeah. I
So
agree. Mhm.
yeah, and I was gonna say, yeah, I think that's fine. There's like a way that I learned myself, like figuring it out like a while ago that would let you get the exact value, but essentially, yeah, I would do it that way. I
Okay.
would think about like the perfect squares that it's in between, and then be able to rule out stuff that are not within that range. And then I can just choose, whichever it's closer to.
Yep.
So, yep. So yeah, essentially here, you read it and there's no friction, so we don't have to deal with that. But yeah, you did what you were supposed to do, which is you just take inventory of the values that you're given, and then you decide, what type of kinematic formula to use if they give us, this information. So, yep, not that bad.
Yeah.
Mhm.
I just have to remember my formulas better.
Yeah,
Yeah.
yeah, it can help to like, I guess, understand that if you're adding like things, they must have the same unit. So like, v squared would be like meters squared over seconds squared, right? So then, a times distance, so acceleration is meters per second squared, times distance, which is meters, would give us meters squared over second squared, which is that, right?
Yeah.
Whereas here we have, well, velocity meters per second times time, which is second, would give you meters, and that's why it would be equal to the x. And then same thing here. This has to be something to give us meters at the end. So like acceleration meters per second squared times time squared, which is s squared, gives you meters.
Oh, I see. This is a cool trick. I
Yep.
like it.
So yeah, if you can only add things if they have the same units. So yeah, so that can help you figure out, if it is going to be like the AT squared term or like the AX term. The 1/2 and the 2 are, you just kind of have to remember that part because there's, no way to kind of like figure that out directly. But yeah, this should help you just remember which one will have the AX term and which one will have the AT squared term. Yeah.
Yeah.
Mhm.
Sounds good.
Yep. Yep.
Oh boy. Okay. Yep, we did this one. All right. Which of the following statements is not true concerning the element that it describes? Um, so I think it has to do like with atomic trends, like periodic table trends.
Okay.
Um, lithium has a second largest atomic radius in period 2. And then I was struggling with something very basic, like period 2 is referring to a row, right? Yes. Yes. So lithium, does it have the second largest atomic radius? So atomic radius, as you add more electrons, you should have a larger radius, right? But you have the competing force of like the protons in the nucleus. And so if you have a high proton number, like you're also, so I think it decreases down column.
So
You're adding protons.
way you're gonna see an increase in atomic radius,
So it increases down
and to the left. Uh-huh.
the left.
That's the
So
only
this is
periodic. Yeah. Sorry.
okay, I see.
Yeah, like every other periodic trend is this direction.
The electronegativity one, yep.
Yeah, that'll be electronegativity, electron affinity, ionization energy. But the only one that will be in the opposite direction will be atomic radius.
I see.
So
So yep, so lithium, uh,
what was it? Let me see the trace again.
lithium has because it's in periods that they're in period 2, second largest atomic radius in period 2. So that arrow that you showed me, it was decreasing or increasing? Sorry.
Oh, going down and to the left, you're increasing in atomic radius.
Increasing. Um, so you should have the highest atomic radius with lithium. If that's in period two,
Okay.
right?
So let's see.
Instead of second largest,
Uh-huh.
you should have the largest, right? If you're increasing down and to the left, lithium should be the largest,
Mm-hmm.
not the second largest. So I guess that would not be true.
Okay.
We could go over the others.
Mhm.
Iodine has the highest electronegativity in period 5. Iodine. So electronegativity is this way, increasing. So in period 5, um, yeah, I would say it has the highest electronegativity. Yeah. Do you agree?
Tell me about the electronegativity periodic trend.
Yeah, so it's increasing, it's like a diagonal increasing highest at fluorine, like this. So in period 5, um, I'm only comparing period 5, so I think that it would be the highest in period 5.
Okay. So yeah, let me see those choices again. Iodine has the highest electronegativity in period 5. Um, okay, sure. Maybe, I mean, I don't know if we're supposed to consider the xenon or not,
I
but
don't know. I guess that's also a question I have, like, because I've only always like dealt with like electronegativity questions, usually in a group, not in periods. And it's usually like we never like discuss like the noble gases. Um, yeah.
go on with what you're saying.
Yeah. So like I would also have that question of like
Mhm.
what's like the electronegativity trends with the noble gases.
Got it. So, okay. So I guess we evaluated A and B. What about C and D?
Iron is a transition element. Groups. Um, it's a metal.
Okay.
I think so. I think it's right here. But could we review this? So I know we have the alkaline earth metals and alkaline metals. The alkaline earth metals and alkaline something else. Uh,
Alkaline metals, alkali earth. Mhm.
yes. We got our transition metals, right?
Yeah, here.
Yeah. So yeah, and then you have this is where you have like the metalloids.
Mhm.
Then you have your like halogens, or what do you call them? Your non-metals. Non-metals?
Oh, were you asking about this one or
Like
just
all of them, like right of the metalloid ladder. Is it just non-metals, I think?
Hmm?
Yep. Yeah. Yeah,
Yeah,
halogens and the noble gases, yes.
yep, yep.
And yeah. And these are metals too, I think. 58 and the last two that yeah.
I like the lanthanide, actinide.
Yeah. Yeah.
Okay.
Um yeah, so I think iron is a transition element.
Yep.
Neodymium is a rare earth element. I have no idea what this is. Um I've never heard of it. So maybe it is rare, but I have no idea.
Mhm.
Uh, yeah, that's my thought.
Okay. So, um, the lanthanides are rare earth elements. So neodymium is in that over there. Yep.
60? Okay.
So, okay, so now I guess it comes down to, so is iodine the most electronegative in period 5, or is lithium the second largest atomic radius in period 2? So since its radius increases from going down into the left, um I think that should be our,
Yeah, I agree. Yeah.
yeah.
And I guess for electronegativity, like the noble gases would not have like the highest electronegativity. They would have the highest like ionization energy, right? But they wouldn't, right?
Yeah,
Like to
so
remove it would be a lot harder. Yeah.
yeah. I think that can be a little weird because I remember taking like a class called inorganic chem, and then they told us in that class that neon was the most electronegative, or helium, it was something like that. But yeah, I don't know if that is going to be a thing here. So yeah, between A and B, like either B, which would mean that the xenon after it has a higher electronegativity, or lithium. I guess the answer would be A, just because lithium should have the highest or the largest atomic radius in period 2.
Okay.
Yeah.
So it's just making because I think that's good because I'm always like thinking in terms of like what's the greater pull, like Z effective when I'm answering radius questions. But just knowing that like the trend is increasing in the opposite direction of like the electronegativity difference or like all the other trends helps.
Yeah.
Yeah. Okay.
Mhm.
How much heat is produced from the complete combustion of 30 grams of methane if the enthalpy of reaction is -890 kilojoules per mole? Heat. So I am thinking of the equation Q equals MC delta T. Um, 30 grams of methane. Um, I think it's um C. Do we even have C? We have enthalpy of the reaction, which is H. What does that even
Okay. So
mean? We're not given C.
correct. So what we can do is we can look at the units here. So we're given mass of the methane, and then we're given the enthalpy, which is in energy per mole, and the answers are all in joules. So we can essentially just figure out how many moles is 30
Oh,
grams of methane,
yeah,
and then figure out how many whatever that and energy would be.
I see. So we just have to convert 30 grams of CH4 to moles. And from that, just multiply it by the -890, and that will give us kilojoules.
Mhm. Yeah,
Um, carbon
16.
carbon is 12. Yeah, and for hydrogen, that's 16. -890 divided by,
This would be roughly 2 moles of the methane, right?
yes, that's correct.
Okay. And then we can just, you know, I guess multiply this by the 2 moles. So that should be, this is negative or whatever. Let's say 900. kilojoules per mole, multiply it by 2 moles, you would get 1800 kilojoules, which would just be, well, 1800 times 10 to the third power for joules. And, the choices have, that
Yep.
number, as, in 2 digits. So I'm going to change this into 2 digits. So I always, whenever I do that, I say this out loud in my head because it's easy to mess up. So like if I'm going from 1800 to 1.8, I say left subtract, right add. So I'm subtracting or moving the decimal point to the left 3 times to get 1.8. And so I'm going to add on the right side 3 times to get times 10 to the 6th power.
You said left subtract, right add,
Yep.
and you moved it to the left, no?
Oh, sorry. So basically, all right, so if I'm going from 1800 to 1.8, on the left side, I'm reducing that number. So I say left subtract and then right add. So on the left side, I'm going from 1800 to 1.8, which is moving the decimal point over to the left 3 times. So on the right side, I need to compensate for that. So I add the 3 decimal points to get times 10 to the 6th.
Mhm.
So, so yeah, just from like this calculation, you can already get rid of C and D because that has 4.7 in the front.
Yeah.
And so, the 1.8 that we got is closer to the 1.7. So yeah, we can get rid of C and D. And then it would be choosing the one that has the correct exponent. And that'll be B.
Yep, I agree.
So yeah, so you were good in that you were thinking about the Q equals MCAT type of equation, which is what I was thinking of as well. But as as you said, we don't have specific heat for methane, right? Like the only one that we know is like water. So and we don't know the temperature change either. So that equation's not going to work. But if we don't know what equation to use, we can use our knowledge of units to figure that out, and vice versa. So if they gave us the mass of methane and they give us the enthalpy, which is energy of like per mole, we can take the mass of methane, convert it into moles, and then use the enthalpy of the reaction to figure out what the energy would be for the 30 grams of methane.
Yep, I see.
Yep. So yeah, so sometimes if like a formula that you're thinking of doesn't work, you can still figure something out based on units.
Good. Yeah. Good. I think we um
Mhm.
are done unless
Mhm.
yeah, I think we're done. Are you okay with being done now?
Yeah. I'm wondering, I just want to take a look at some other like of the discrete questions that you may have skipped or um something like that.
Probably, especially near the end. Um,
How did you feel about that one?
um, I think I chose this one.
Uh-huh. What tell me your like reasoning?
It's bulkier, for SN1.
Uh-huh.
Um, so
So
I remember like SN2, like SN1 would be preferred, like if it's like a bulkier substrate. Mm.
yep. So yeah, so this would look like, let's see, like propane, 1, 2 chloro, 2 methyl propane, right? It's
Yep.
like a tertiary structure, and that is what would make an SN1 reaction be more likely.
Yes. Yes.
So, because yeah, this is gonna leave, and then you'll get this tertiary carbocation, which is the most stable type of carbocation, and then yeah, SN1. So good job, awesome. So okay, so I guess you will continue doing the blind review for this test, and your next test, um, will that be on
Next Saturday.
16th?
Do you have a recommendation for which test I should do?
So you've done blueprint full length. You've done, let me see, blueprint full length 5 and full length 3, right? So which ones have you done, like blueprint full length 1, blueprint full length 2, blueprint full length 4?
No, I haven't. I've just been going in a weird way. Yeah.
Okay, okay. Uh yeah, so I would say do any of the blueprint ones. But for cars, yeah, I want you to do the cars section too. So, but not the blueprint cars. Either do one of those Jack Weston cars exams, or choose like 53 or 9 passages from like AMC cars, like either the question packs or the diagnostic tool. Just because, just because for this test, like, you know, um, if you didn't do the cars section, then it kind of makes it so that you have more energy for like BB and Psych Soc. So I just want to make sure that we, you know, have you do every single section to make it the most accurate like simulation of the test. So yeah, I would say do any other blueprint tests, but just switch over to Jack Weston, one of like the cars exams, you know what I'm talking about, right? Like Los Angeles
Yeah,
stuff.
I think I'll do that. The reason I didn't want to do another one is because
Yeah.
I still haven't reviewed like my like AMC cars one, and I just felt bad about like doing more without like reviewing them. Yeah. Mhm.
Yeah, yeah. No, I understand. But it seems like, you were doing pretty well in that. Um, but yeah, we just want to, because it's like cars is a little different. It's not like something that you learn from cars may be asked again or something like the science section. So I think cars, you will still benefit from doing cars even if you didn't do like your blind review or regular review. So I would just add that in, just so we get accurate results for the sections that come after.
Yeah.
So yeah, I would say so yeah, continue doing your blind review and regular review of the test of the blueprint full length 3. And on Saturday, the 16th, you'll take your next blueprint test. And yeah, just do the cars section from Jack Weston or AMC. Um, did you want to meet this week?
Yeah, I thought I wasn't going to have any energy, but it worked out when we met on Wednesday. So we could do like Wednesday or Thursday,
Mhm.
and for like an hour, like 7 to 8, like
Mhm. All right.
after work.
Yeah.
Yeah.
Okay. So let's see. So Wednesday, March 13th at 9 at 7 p.m. you said?
7 p.m. my time.
Oh,
Uh,
you're in Philadelphia? Yeah.
yeah.
So yeah, uh same thing, like same time zone. So oh crap. All right, so I put you in for Wednesday the 13th at 7 p.m. Eastern.
Sounds good.
Yeah, by then you will probably have a lot of questions from the blind review and review of the blueprint test that you just did. So yeah, so we can go over that stuff. And then on, yeah, Saturday you'll take your test, and that's all. That's that, right?
Yeah. I am like guess approaching being exactly 1 month out, should I start doing like the section banks for the like AMC material, or
Uh, let's see.
should I keep doing UWorld? I mean, UWorld is working, so I could just keep doing that.
Yeah. I think like as long as you are getting like the blueprint tests in, that's good. And um I think I wrote for you. I think I wrote that you are continuing third because I think you did all of the AMC ones, all the AMC practice tests.
A long time ago, but yes, like last year.
Okay. Okay, maybe I can add that in, but from what I see, it's going to be more third party full lengths and perhaps the week before we can just have you do like an AMC exam. Um, but yeah, in general, you're if you are finished with doing your review of the test, you can kind of like see like what types of areas that you feel like you need more work on, and then you can use UWorld to like populate, passages about that topic. And in addition to like, have you done the AMC section bank before?
Yes. Yes.
Okay. So yeah, I would, I guess, prioritize the UWorld over that. Just because, you know, you've done it before. I know that you've done it a long time ago, but if it's one or the other, yeah, I would say UWorld. Just because just because you haven't done them before, and you can also just choose which topics to uh do UWorld stuff on.
That's good. Well, thank you.
Yep.
This is helpful. And you're wearing orange glasses, right?
Oh yeah, they're like red.
Oh, yeah, I see.
Yeah. Because yeah, why?
Yeah.
Oh yeah,
You're
so
wearing your glasses. Yeah.
because like I'm in front of like my computer mostly all the time, so you know, I mean, there's like like how there's blue light blocking glasses. Um, so that's a thing because ironically, so like, have you ever heard of like blue light therapy? So sometimes like if people have seasonal affective disorder, right?
Yeah.
Uh,
Like light therapy. I didn't know it was called blue light therapy.
yeah, because it has to be blue light because blue light once it hits our retinas, it causes like something like retinol to turn into like an aldehyde. Um, but what it does is that it can release serotonin, so that's good for your mood. So that's why it's used in to treat like seasonal affective disorder. But serotonin is also something that promotes wakefulness. So like blue light that's given off by our computer screens and stuff can interfere with our sleep. So I have like an app or whatever called Flux, and it's F period LUX, and it automatically like you put in like your wherever you live or whatever, and like automatically as the sun outside goes down, the amount of blue light that your monitor will show goes down, and the more red light will be shown. So I have like a program that does that, and but it wasn't enough, so I have these glasses because now instead of just blocking blue light, like it takes all like light that I guess hits my glasses, and it puts a red tint on it, so it makes it a lot more easier. Yeah, it can help
Yeah.
like the um yeah, it can just yeah, so it can just help with the intensity and of all that light hitting your eyes and making it dry and making it painful and things like that. So yeah, it's just a combination. It's like blue light blocking glasses isn't enough. So I have the ones that are just tinted red to help
Mm,
that out. So
okay. Sounds good.
yep.
Yep.
But yeah, if you feel like you are getting too much brightness or whatever from your screen and things like that, I would recommend something like Flux and, of course.
I have some orange glasses lying around. I might.
Mhm.
Yeah.
Yeah, that can really help. So yeah, that's why I wear these, and I mean, yeah, and yeah, it's pretty cool, I think. But yep, so that's why I have them. So yeah, any other questions?
Nope. I'm good.
Awesome. So yes, good luck. I'll see you on Wednesday, okay?
Yep. Sounds good. Bye.